Hi everyone, my name is Chau. Today I'll be presenting on carbapenem antibiotics, which are the last line of broad-spectrum beta-lactam antibiotics, a very important group of antibiotics used in modern medicine. So for structure and chemical properties, carbapenems are a type of beta-lactam antibiotics, meaning they contain a beta-lactam ring as their core structure, which is essential for their antibacterial activity. Their structure are shown in the diagram below, where beta-lactam ring is located here. A four-member, highly strained cyclic amide with three carbon atoms and one nitrogen atom that act as the core structural component of key antibiotics like penicillin and carbapenems. However, unlike penicillin, carbapenems have a five-member carbapenem ring that replaces the sulfur atom, making them structurally unique and more stable against many bacterial enzymes. They are considered broad-spectrum antibiotics, meaning they are effective against gram-positive bacteria, gram-negative bacteria, and also anaerobic bacteria. Another important feature is that they are resistant to most beta-lactamases, which are enzymes produced by bacteria to destroy antibiotics. Carbapenems are also water-soluble, so they are usually administered intravenously, and they are unstable in their natural form, such as tianamycin, which will easily degrade. Some common examples include imipenem, meropenem, ertapenem, doripenem for the carbapenems. For the source and origin, carbapenems are semi-synthetic antibiotics, meaning they are chemically modified from natural compounds. They are originally derived from Bacterium Streptomyces cattleya. The first discovered compound was tianamycin, but it was unstable, so scientists chemically modified it to improve its stability, antibacterial spectrum, and resistance to beta-lactamase enzyme. So this led to the development of modern carbapenem drugs. And for the mode of action mechanism, carbapenems are bactericidal antibiotics, meaning they kill bacteria rather than just inhibiting growth. They work by targeting penicillin binding proteins, also known as PBPs. These proteins are involved in building the bacterial cell wall. So specifically, carbapenems inhibit the transpeptidase enzyme, block peptidoglycan cross-linking, and also weakens the bacterial cell wall, as shown in the picture above. So as a result, the cell will become unstable, leading to cell lysis and bacterial death. They are effective against bacteria such as E. coli, S. aureus, and also Pseudomonas aeruginosa. And for their resistant mechanism, despite their effectiveness, bacteria can develop resistance to carbapenems. One major mechanism is the production of carbapenemase enzymes, which break the beta-lactam ring and inactivates the drug. For example, Klebsiella pneumonia produce the KPC enzymes that destroy carbapenems. Other resistant mechanisms include efflux pumps, which actively pump the drug out of the cell, foreign mutations that reduce drug entry into the bacteria, and also alter PPPs as mentioned before to reduce the drug binding, as shown in the picture above. Microbes that show carbapenem resistant include carbapenem-resistant Enterobacteria ca, CRE, including E. coli, Klebsiella pneumonia, Klebsiella oxytoca, Enterobacter aerogenes, and also Enterobacter cloacae. These bacteria are of particular concern in hospitals because they are difficult to treat and can spread resistance to other bacteria. So, for the medical uses and importance, carbapenems are used to treat severe or life-threatening infections, so they are often considered as a last-line antibiotics, meaning they are only used when other antibiotics fail. So they are used to treat diseases such as sepsis, pneumonia, urinary tract infections, intra-abdominal infections, and also multi-drug resistant infections. However, like all drugs, carbapenems have side effects too. The common side effects include nausea, vomiting, diarrhea. The more serious side effects include neurotoxicity, such as seizures, especially with imipenem, and also allergic reactions, especially in patients allergic to penicillin. So, what's the importance in modern medicine for carbapenem? Is critically important because they are used to fight multi-drug resistant bacteria. However, overuse can lead to the emergence of carbapenem-resistant Enterobacteria ca, which are very difficult to treat. So this highlights the importance of antibody stewardship, where it's a set of coordinated evidence-based strategies designed to promote the responsible use of antimicrobials to ensure patients receive the right drug at the right dose. So for the cost and accessibility for carbapenems, one example is the meropenem, which costs approximately 40 ringgit to 150 ringgit per vial in Malaysia. The price is based on the conversion from US dollar market to Malaysian ringgit. So this high cost is due to complex chemical synthesis and resistant clinical use. So in conclusion, carbapenems are powerful broad-spectrum antibiotics which are highly effective against the resistant bacteria, but must be used carefully to prevent the resistance. So this was the reference. Thanks.